Today we are going to discuss two ways by which evolving bacteria like Staphylococcus aureus can get new genes. One way is via mutations. Mutations are heritable changes in the genetic material of an organism. Mutations can be silent mutations, meaning that no observable change results from the mutation. But mutations can also lead to new physical characteristics. One way that mutations arise is from point mutations, where a single nucleotide in the DNA sequence gets altered to a different nucleotide base. If this change results in a change in the amino acid sequence, this is called a missense mutation, which can possibly result in an observable change in the organism. There are also other types of mutations, like frameshift mutations, in which one or more nucleotides are inserted or deleted in such a way that the codon reading frame gets shifted, and nonsense mutations, in which a premature stop codon is introduced. These types of mutations are more likely to result in an observable change in the organism because they are very likely to yield the gene product non-functional. S. aureus can also gain changes in its DNA via recombination, a process by which DNA gets exchanged between two organisms. There are three ways this genetic recombination can occur. Transformation describes the process in which free DNA released from one cell gets taken up by another. Another form, conjugation, is when DNA transfer involves cell-to-cell contact and a conjugative plasmid in the donor cell. This conjugative plasmid is termed the F plasmid, and F+ cells can transfer this plasmid to F- cells. The final type of genetic recombination is transduction, the process by which DNA is transferred via a bacteriophage. When bacteriophages infect bacteria, they trick the bacteria into reproducing their genetic material for them, and they then repackage their material to go off and infect another cell. Sometimes in this process, the bacteriophages can accidentally pick up some of the bacterial genome, and then they will also transfer these bacterial genes between bacteria. This video discussed methods of horizontal gene transfer between S. aureus of some of the same generation. Mutations can result in new combinations of genetic material that may or may not benefit the organism. Recombination can result in the transfer of genetic material between two organisms of the same generation. These processes can lead to new physical characteristics that may help the bacteria S. aureus survive better and evolve to live in their ever-changing environment.